back.
Okay, and now I'm gonna go back to giving you the host. All right, so
Jessica, it wouldn't hurt me to hear you say again what you just said.
Okay.
And it'll work for archival purposes.
Yes.
Steve knows I need the help.
Let me just start over. I'm going to start with a quote from the New York Times. This was an article that appeared in the science section about 3 months ago. It is entitled, "Odd Clue from Deep in the Sea." Quote, "A bizarre tentacled microbe discovered on the floor of the Pacific Ocean may help explain the origins of complex life on this planet and solve one of the deepest mysteries in biology." The key word there is complex. Remember that. Let me go on. What is the mystery that this finding solves? The mystery is as follows. We know that life arose on Earth approximately 3.5 billion years ago. I talked about that ultimate origin in a previous lecture. The subject of this lecture is the fact another occurrence that occurred 2 billion years ago. In other words, life arose 3.5 billion years ago, and during its first 1.5 billion years, life evolved into 2 classes of organisms. Those 2 classes are the bacteria and the archaea. But then something happened approximately 2 billion years ago. In other words, life had been already around for 1.5 billion years, and then something happened 2 billion years ago. And that something is the origin of a new kind of cell, a cell that was much more complicated than the cells of either bacteria or archaea. And I will go into those details and explain what's going on to the best of our knowledge and how this finding of the odd clue from deep in the sea has given us some real insight as to what happened. Complex life, this thing that happened 2 billion years ago, is all the life that we know now other than bacteria and archaea. We call all these other kinds of life, animals, plants, fungi, algae, etc. We call all of those kinds of life eukaryotes. That word is eukaryote. And that word eukaryote means true cells, complicated cells, the kind of cells we have in our body and the kind of cells that plants and animals that we see on the earth have. Okay. This happened 2 billion years ago. And that is the mystery, and that is what this talk is really all about. We are multicellular life. Plant, fungal, animal, we're all called eukaryotes. Eukaryotes, the kind of organisms that we are, for the first time in the history of life, were able to form multi-celled organisms. The first multi-celled organisms were probably looked a little bit like sponges. But even sponges have some parts of them that do one activity and other parts of them that do another activity. And of course, animals like us have all kinds of parts that do all kinds of activities. But in the bacterial world and in the archaeal world, that never happens. Bacteria can form colonies of hundreds or even thousands of organisms. But these colonies are just collections of separate organisms. They are not differentiated so that one part of the colony does one thing and another part of the colony does another thing. Once the eukaryotic cell formed, it was able to do that. It was able to form colonies in which parts of the colony did one thing and parts of the colony did another. So let me describe for a moment what bacteria are like. We all know about bacteria, and they are one of the original forms of life that stemmed from a common ancestor.
Excuse me, Jessica, do you have a presentation going? Or no?
I don't know what you mean.
Do you have any slides?
No, I sent them out on an email. I do not.
Okay, so we should go back and look at the material I sent out to everybody.
That is correct. Yes,
Okay.
Thank you, Bob, for
I
saying that.
just wanted, if you had a presentation, we weren't looking at it, and I just wanted to make sure that was the case. Thank you.
Yeah, thank you. Okay, so going back, let me describe for a moment what a bacteria is like, or a bacterium, to use the singular. If you look inside a bacterial cell, it's relatively formless. There's not a whole lot you can see with a light microscope. Bacteria have just one chromosome, typically with about 1,000 genes. Their chromosome is circular. It is attached to the inside of their cell wall. It's made of DNA, just like our chromosomes are. It has information encoded in that DNA, just as our chromosomes do. And it works pretty much exactly the way our chromosomes do. But in the bacterial cell, there is no nucleus. In eukaryotic cells, there is. And the chromosomes, most of the chromosomes of eukaryotic cells are in the nucleus. That's not the way it is in bacteria. But the one thing that bacteria do, all bacteria, is that they are very good energy producers. They have evolved ways of breaking down sugar very efficiently to obtain energy from the breakdown. As a result, bacteria, generally speaking, divide and multiply very quickly. The E. coli that lives in your gut, for example, under good circumstances, can divide every 20 minutes. So understandably, shortly after the beginning of life on Earth, within 3 billion years ago, bacteria had spread all over the oceans. During that early period, some bacteria, in fact, evolved to become parasites on other bacteria. The first viruses, the so-called bacteriophages, evolved long ago, before there were ever eukaryotes on the surface of the Earth. Viruses are nothing more at that time than bacteria that had lost a lot of their genes, retaining only the ability to attach and parasitize other bacteria. I just thought I'd add that in view of the coronavirus out there, because frankly, bacterial viruses act just like animal viruses do, and they parasitize other species. But getting back to bacteria. The bacterial cell then is, to summarize, a formless bag. It has one chromosome. The chromosome acts like our chromosome does. Bacterial chromosomes make RNA copies of themselves. The RNA copy of themselves goes off to form various proteins, just give the information to other parts of the bacterial cell to make various proteins. All of that acts just like our cells do. But in the bacteria, those processes are just in the whole cell. They're not regulated to one particular part of the cell or another. Now, Archaea, the other kind of organism that existed back then and still exists today, they're also single-cell organisms. But Archaea never evolved very efficient energy production mechanisms. So Archaea grow very slowly. The Archaea that we're going to be talking about later in the talk takes 25 days to multiply, to divide one cell into two, as opposed to 20 minutes for a rapidly growing bacterial cell. Archaeal cells, when you look inside them, have what we call membranes. You can see them. They have like little bags inside them in which certain processes take place. They're much, if you look at them with the light microscope and you have stained them appropriately, it looks like there are all these little chambers inside an archaeal cell. Bob Corsan gave me a wonderful presentation about the folding of proteins. And one of the important things about the folding of proteins is that they fold in water. And the way they fold alters their activity. And when a protein lands on a membrane of one kind, it will fold one way. And when it lands on a membrane of another kind, it may fold differently. And so having membranes gives the archaeal cell a much more expanded ability to do various chemical processes. So up until 2 billion years ago, we had these two kinds of organisms. The bacterial cells, which could process energy very, very quickly and get energy very, very quickly from burning sugar. And the archaeal cells, which could make all kinds of different things, but very slowly. And really, most archaea have to live near thermal vents and things like that because they never did evolve a good energy producing mechanism. I'm going to skip over a description of DNA, RNA, and protein formation. It's on the outline that I sent you. It's the third picture that I sent you, and it's just an outline of how the DNA code works. And for those of you not very familiar with that, let me just simply say that the DNA in all organisms that use DNA to encode them is the hard disk. RNA is the copy of the hard disk that is sent to the central processing unit. The central processing unit is whatever in the cell takes that code and translates it into protein. Proteins are the program for this analogy that appears on the screen. Proteins are what counts. And you can look at that or read up on that separately. Now, bacteria and archaea all do this translation from DNA to RNA in the same way. And it turns out that we, eukaryotes, do it the same way. So it became obvious to researchers, to biologists, that basically we all, bacteria, archaea, and eukaryotes, stemmed from the same common ancestor. But how did that happen? How did that happen? There is a phenomenon in life, in all of life, in life that exists in the world today, called symbiosis. And when you have symbiosis, two different life forms end up growing together, one inside the other. You've all seen lichens growing on a rock. Lichens are actually two separate organisms growing together. One of those organisms is a fungus, and fungi have the ability to grow on all kinds of surfaces, and they have the ability to make all kinds of proteins, and they have the ability to derive minerals from the rock's surface. Algae, green algae, which is the other kind of life in this symbiote that I'm using as an analogy. Green algae can photosynthesize. That is to say, out in the sunlight, they derive energy. Those two organisms get together to form what we call a lichen. The algae cannot grow on the rock by itself. The fungus, which can grow on the rock, cannot derive energy from the sunlight. The fungi have to derive energy from sugars in the soil. And on a rock, there are no sugars. So the fungus could not grow on the rock alone very quickly. It can grow slowly. And it would have to send down tentacles into the ground to get its energy. And the algal cell couldn't grow on the rock at all. But you put the two of them together and you have an organism that can derive its energy from the sunlight and grow on the rock. That kind of symbiosis exists out there in nature even today. And so it became obvious, or it seemed to be obvious to the researchers, that the original eukaryotic cell was some kind of a symbiotic organism between a bacterial cell that gave it its energy production capacity and something like a fungal cell, but it was an archaeal cell that had a good ability to make all kinds of complicated proteins, but slowly. And that made sense. But we didn't know, how do we prove this? How do we find that original organism? Now, 10 years ago, there was a Japanese researcher who was studying what are called Archaea, we've talked about, and he found a particular kind, he was studying a particular species of Archaea called Promethoarchaeum. Don't worry about the name. And he tried to grow this in the laboratory. And what made this particular Archaea especially interesting to researchers is that when they did a genetic analysis of the chromosomes in this archaeal cell, they discovered that it had 3 genes that were, prior to this discovery, only found in eukaryotes, never found in bacteria. So they thought, goodness gracious, this archaeal cell has 3 genes that we have, that all eukaryotes have. We have those 3 genes in our body that are found in this archaeal cell. They don't do the same thing in our body that they do in the archaeal cell. But it makes sense. If we were formed from a symbiote in which this species or something like this species was the archaeal component, then yeah, we would have those genes in us, and we do. Now, this particular archaeal cell grows in many, many riverbeds, basically all over the world. It's quite common, very hard to grow. Took this man 10 years to get it to actually grow and to actually take a picture of it. And that picture is the subject of the article. I hope it came across as a copy to all of you. And that picture shows 3 or 4 of these archaeal cells, each one of which has huge long tentacles coming out of it. And if you look closely, you will see little round balls occasionally on those tentacles. Those little round balls are bacteria. Those tentacles are the archaeal cell. And what's going on in that picture is that the bacteria are, if you will, feeding on all the proteins that the archaeal cell is making. And the archaeal cell is deriving energy from that bacterium. That is to say, that bacterium is breaking down sugars, which are then being absorbed by the archaeal cell for its purposes. This is indeed an illustration of what happened. So the conclusion is the original common ancestor of we eukaryotes was the ancestor of Promethoarchaeum. Of course, that ancestor has evolved over 2 billion years to give us Promethoarchaeum. But way back then, 2 billion years ago, a symbiote was formed. And that symbiote was the original eukaryotic cell. And as so often happens in evolution, the fact that it had membranes inside the same cell as it had efficient energy production gave it, as a side effect, the ability to divide quickly, and secondly, to form a multi-celled organism, and thirdly, to form a multicelled organism in which one part of the organism did one thing and another part of the organism did another thing. That was an accident. It just happened. The original eukaryotic cell could not have predicted that. So in summary, there was life before the eukaryotic Big Bang, and that life was bacteria and archaea. And then as symbiosis occurred, eukaryotic life arose, eukaryotic life exploded to cover the world. And that's the end of my presentation. I don't know if it made any sense, but thank you for listening.
Thank you.
Thank you.
Can we ask questions?
Of course.
Well, I've written down 3 questions. The first one is, you talked about bacteria. Its food or fuel, shall we say, are sugars that it ingests or takes in. Where does it get the sugars?
That is a very good question. The sugars today come from all kinds of life in the sea that dies off. Where did they come from originally? They must have come originally from sugars that were present in the primordial sea. I imagine there were sugars there because we found carbohydrate-like substances even in meteorites. So I imagine they were present in the original primordial sea.
So
That's the
sugar
best.
sugar is a form of, is sugar life or animal in the animal kingdom, or is it in the mineral kingdom?
Sugars are chemicals that are called carbohydrates. They have carbon, hydrogen, and oxygen. And, you know,
currently sugars are in say plants, right?
Yes.
And
yes.
so the bacteria are feeding on plants and sucking out the sugars in effect.
Well, the plant dies and the sugars are liberated out into the environment, but in essence, you're right.
Okay, so it's after the plant dies. It's not while the plant is still alive.
In some cases, it is.
Because what I'm getting at here is that life, including plants, is composed of bacteria also. Eukaryotes have bacteria within them, don't they?
Yes.
So bacteria living on eukaryotes is in effect bacteria living on other bacteria.
Well, not exactly. Let me back up for just a moment. The original eukaryotic cell, which was a symbiote, had bacteria inside it. And today, our cells have those descendants of those bacteria, which we call the mitochondria. Those are descendants, and mitochondria have their own genes. They're in our cells. All of our cells have mitochondria. But the nucleus of those cells, 12 genes, is still in our mitochondria. It's not in our nucleus as are all our other genes. Interestingly enough, mitochondria are inherited from your mother. Father does not contribute mitochondria to you. But that's basically an aside. But bacteria are separate kinds of life. And back in the early days, there were sugars, carbohydrates, free in the primordial soup that they fed on.
Okay.
Make sense? Your other two questions?
Oh, I don't want to hog the show here. I actually wrote down mitochondria because I've heard of it, and I am aware that mitochondria is an ancient life that lives within all of our cells, but it's separate. And I wondered if you could, I mean, I think of it kind of as, you talked about little bags. I think of it as a little bag within our cell. And I'm wondering if you could mention how mitochondria interacts with whatever else is inside the cell, such as the nucleus.
Okay. Mitochondria are that one of your cells has maybe 50 mitochondria in them. And these mitochondria all have the same gene that you inherited from your mother, same genome. And what they do is they break down sugar. And as they break the sugar down, they release energy from the breakdown. That energy is processed in a complicated mechanism I call ATP, and adenosine triphosphate, adenosine diphosphate, and things called the Krebs cycle, and that would take me hours to explain. But the bottom line is that they act for our purposes just like little bacteria living within us. And that is exactly what they are. But the only thing they do is they break down sugar and get energy out of it. And they are called the mitochondria.
And my last question was, you talked about bacteria as the cell wall of the bacterium interacts with another cell wall, it causes it to bend. And the different, if it interacts with one type, it'll bend in a certain way, and another type it'll bend in a different way. And of course, bending is just movement. So it's that's what I wanted you to talk about, a little more about the different, what is going on between the two cell walls that creates the different bending. It's a different type of energy or a different amount of energy or something like that.
Okay, let me back up. Yes. And first of all, the organism, the primordial organism that had cell walls was an archaea. The bacteria only has an outer cell wall, does not have membranes inside it. The archaeal cell does. Now, these membranes have various collections of protein, fat, and carbohydrate in their structure. So if you took two different membranes from two different, say, parts of the archaeal cell, they would be a little bit different. One would be made out of a lot of a protein that, let's say, has a lot of alanine in it, which is one of the components of some proteins. And the other one might have a different protein that has a lot of serine in it, which is another amino acid. So those two proteins are different. If you drop them on a glass surface covered with water, they would fold differently. On the other hand, if you have one protein and you're dropping it down on two different surfaces, it will fold, but it will fold differently when it drops on one surface than when it drops on the other surface. The reasons for that are the interactions between the various charges on the protein and water molecules, and the interactions of those water molecules with the surface. And it's actually very interesting, and in a way, as that article that Bob referred me to explains, if you get through all the chemical hogwash in there. A very simple process. Water, because of the way water molecules act, actually has structure. You don't think of it that way, but it does. And the structure changes as it is on one surface versus the other. And when you drop down on it, a rubber band or a protein that has the tendency, because of its charge structure, to fold, it's going to fold differently on different surfaces. That's all. So the archaeal cells had that. They evolved that. They were able to make many, many different chemicals, proteins, than the bacterial cell could. But they did not ever metabolize very well. It took them a long, long time to grow and to do all this kind of stuff. The bacteria, by contrast, could make different proteins and did, and had 1,000 genes that did so, but it did not have the capacity to form anywhere near as many different structures as the archaeal cell could. Put the two together, and you've got a magic organism. That's the eukaryote. Okay, does that make sense? I see you smiling, so maybe I helped.
Oh, no, it's fascinating. It's just fascinating. Thank you.
I have a question that's related to the question that you just discussed.
Yes. I can't hear you, Charles.
Charles, you're on mute.
Can you unmute, Charles?
Yeah, I'm okay now. My
Okay.
wife told me that when I use, if I put my phone, if I put it on mute, I can press the space bar and I come off of mute.
Oh, wow.
So I'm off of mute now.
That's right.
Okay.
So let's see here. Anyhow, I'm a tennis player, and I became interested in how the sarcomeres in the body work. And it turned out that sarcomeres are the basic muscle cells, and they contract when you operate your muscles. So the sarcomeres also have a spring-like function, and it turns out that that spring-like function is not in your tendons or your muscles, but it's in the sarcomere itself. Well, it's in the muscle, but it's in the sarcomere itself. So the thing that's in the sarcomere is called titin, and titin is like the rubber band of the muscle cell. And what it does is it they have a proposed model for what is going on when a sarcomere provides the rubber band function in a cell. It folds as you were describing. When it will fold up and become short to.
Hello? Somebody's phone is.
That's my phone. I'll move it out. Hello? Okay, are we back? Okay,
Yes,
I was talking about titin. And what goes on with the longest molecule in the human body, titin, is that it folds. And that's what occurs when your muscles use springs. And of course, I learned that all tennis strokes are involving springs within your muscles. So it's very interesting. But I'm not sure, since they cannot take a video of what goes on with titin when it operates, because it's much smaller than a wavelength of light, it folds up, they believe, but they're not quite certain about how it works. But it sounds that it's very closely associated with what you were saying, in that the joint molecule titin can somehow create very large forces as it operates in our muscles all the time. So have you come across Titan?
To answer your question, no, I have not, Charles, but I'm glad you brought it up because throughout the body, there is this kind of folding and unfolding of proteins. And as proteins, for example, in the basic energy forming parts of the body, there is a mechanism in which ATP, adenosine triphosphate, is formed and then breaks down into ADP, adenosine diphosphate, releasing the energy of that chemical bond. And doing that, it folds up and then unfolds. And they actually have been able to see this with the electron microscope. So again, what you're saying is a very good illustration. This business of proteins folding and unfolding, springing back and forth, is throughout the body, and it's only beginning to be understood.
I would just,
Go ahead.
I would like to just say an author that is prominent in that field is a Canadian author named Herzog, and I can't remember his first name now, but it's Herzog.
Herzog, is it? Or,
Yes.
okay, thank you.
May I ask, oops, I'm sorry.
Go ahead. I'll ask mine next.
Jessica,
Mm-hmm.
the symbiosis that produced the
Eukaryotes.
the eukaryote is a rare event. I mean, it's not something that happens all the time.
It is a rare event, you're right.
So the second one was also a rare event. So how did it start cascading so that eukaryotes are now, commonplace? I mean, there must have been an accident that had happened in the first place. The second one that happened must have been an accident. What started perpetuating this?
Okay. What started perpetuating life, eukaryotic life, was that you had a cell that incorporated inside it 30, 40, 50 little bacteria to produce its energy, along with all the different membranes that it was able to make.
Yeah, but that was rare.
That was rare. It only happened once.
So how did it get some kinetic energy behind it so that it happened a lot?
No, it didn't happen a lot. That eukaryote evolved. That eukaryote evolved over 2 billion years. But we're all from that same one common ancestor. So symbiosis did not occur all along the way. No,
I got it.
We're all descended from that. Probably the first thing that happened is it just became a very efficient divider. But with all these membranes, by accident,
I see.
there arose one of those eukaryotic cells that had a particular membrane on one side of it and not on the other side of it. So that part of the colony had cells in it that did something differently than the other cells in the colony, and so on and so forth. And when you look at a simple organism like a sponge, there's only about 3 or 4 different things, different kinds of cell in that sponge. And you can sort of imagine that the first thing that happened in the course of evolution, and it could have taken millions of years, was the formation of a sponge-like organism. No, symbiosis is rare.
I
It
see.
happens, but it is rare.
Thank you.
Mhm.
Jessica,
Yeah.
you were talking about how the mitochondria DNA was primarily the process for metabolism for creating energy, but I thought that mitochondrial DNA damage was also related to aging. Have I got that wrong, or is that correct?
No, that's absolutely correct. Imagine, Bob, that as you age, you don't produce energy that well. So what does that do to your cells? What does that do to whatever those cells are doing? They fatigue more quickly. Maybe they can't repair protein as well as they used to be able to do, therefore leading to breakdown of muscle and joints, etc. Does that help?
Yeah,
Is that what
Yeah.
you're driving at?
that's what I just wanted to make sure I had. I mean, so it is still related to the ability to create energy, but it slows down as you get aging. So that makes sense. Okay, thank you.
To follow up on that, Jessica, why does it slow down as you age? Are there fewer mitochondria?
That is a very good question. I can't answer it. It could be that there are fewer mitochondria. I just don't know, Bob.
Yeah, Bob, what I've read about is that the tips of the mitochondria get eroded as you get older. And as the tips change, the ability to have your energy produced is slowed down. And if you get enough destruction to the tips of the mitochondria, that causes the main problems. Is that mostly correct, Jessica?
Yeah, I think it is. I'm not sure what you're referring to in terms of tips, but
I think that reminds me of chromosomes, tips of chromosomes.
that could be. I mean, the mitochondria do have, as I said, 12 chromosomes, if I remember correctly, and they are all necessary. And all the rest of the chromosomes that original bacterial cell that formed the symbiote had moved off into the nucleus. Don't ask me how that happened, but it happened. But in humans, and I think in all animals or plants at this point, the mitochondria have about 12 genes.
Jessica, there's a word, telomeres, related to aging.
Yeah, that's it.
Telomeres. You know what that means?
Yeah, the telomeres are involved in cell division, and if cell division doesn't take place properly, the cell dies.
And there's something about them getting shorter.
Yeah, that could be.
You know,
I'm just a dummy ER doctor by training. So I don't remember things like that, but you're probably right, Bob.
I have an easier question. You haven't talked about plants. You talked about mitochondria and mitochondria, but plants have a different ability. They can take the energy from the sun. And
Sure.
and so is there a bacteria in plants?
Yes, there are, and plants have mitochondria just like we do. When the plant is at night, it is not metabolizing energy, it is not deriving energy from the sunlight, but its mitochondria are processing it just as they do in animal cells. We, plants, and animals all stem from one common ancestor.
But
Photosynthesis is a different organelle. Photosynthesis occurs in a different organelle and was something that evolved in plants.
was that a bacteria? Are chloroplasts, were they bacteria at one time?
No, chloroplasts are separate organelles that evolved, probably started out as some complex membranous structures in what became the ancestor of plants, that because of their arrangement were able to do photosynthesis. Photosynthesis is a rather complicated process, but in essence, it is similar to the way all living structures break down sugars. It just reverses the process to build them up.
Jessica,
Mhm.
I'm a little confused. About the bending. Oh, I have two unrelated things to mention. The bending portion. You talked about bacteria bending. Then you talked about proteins bending. So can you?
Yeah, let me back up. Forget bacteria bending. It's the proteins that I talked about as bending differently in different environments.
Okay, so it's now proteins, and proteins are a function of the bacteria, right? I mean, they
Yeah, all cells, whatever life it is, make proteins.
right. Okay, and then it's the protein that is the thing that bends, and the bending causes the energy
No, the bending is
or
a
the movement.
result of the water molecules and the way they interact with the surface on which it lives or which it falls. And the bending is the result of the various chemical. Imagine, if you will, a string. And imagine that on one end of the string you have sticky glue that hasn't dried yet. And in the middle of the string you have another blob of glue. And you have another string that has neither blob on it at all. And you take the two strings and you throw them up in the air and let them fall down to the ground. Those two strings are going to make a different pattern when they fall down to the ground. The string without any glue on it at all is going to string fall down to the ground in various patterns. And the one with two blobs of glue on it is always going to fall down in a certain, it's going to have a lot of different patterns too, but it's going to be very different in the way it falls down than the one with no glue on it at all. Now, keep that analogy in mind and imagine a protein which has like 100 blobs of glue on it, arranged differently from another protein that also has 100 blobs of glue on it, but in a different order. Those two proteins are going to fold up differently. That's what I'm saying.
Jessica,
Okay, that's good.
maybe it would be helpful if you described what a protein was, you know, the linear string of amino acids and before it folds.
That's basically on my outline of the DNA process. You have a long string of DNA which has information in it. A copy of that string, which is called RNA, is made in the cell. that copy goes out leaves the nucleus, goes out into a part of the cell called the ribosome. And in the ribosome is drawn through a complicated ball, if you will. And as it is drawn through the ball, as each triad of nucleotides goes through the ball and out the other end, each triad causes one of 20 different amino acids to form into a chain. So imagine you've got a ball, it's got a hole in the middle of it. The string of RNA, which is a bunch of nucleotides, is pulled into the hole. As it is pulled through the hole, an amino acid gloms down on top of this ribosomal ball. So the first triad to go through, let us imagine, is AAA. And AAA causes alanine, which is an amino acid, to form. That's the first amino acid in the chain. The second triad on the RNA, let us imagine, is CCC. When CCC goes into the hole, another amino acid, say serine, comes down on the ribosome. Alanine was the first one that came down. Now we have serine that comes down. And on the top of the ribosome, alanine and serine are joined together. The third triad that comes through might be CAU or something. When that triad gets into the ribosome, a third different amino acid is joined to the chain, and so on. So that eventually, reading a string of, say, 300 nucleotides, you end up with a string of 100 amino acids. Those amino acids, only 20 different kinds in life, those amino acids have different charges on them. Some are very complicated with a number of benzene rings and things, or sugar rings, I shouldn't say benzene rings. And others are relatively simple. But the way they are arranged in the protein chain is what determines what that protein does. Once that protein is made, it folds. And there we are. That's
And
2 years of DNA study. Yes, Bob.
I just clarify, it folds on these membranes, and,
Yes.
and the same string may fold in different ways if it's on a different membrane.
Exactly, or at different times on the same membrane. Imagine you have a membrane with a hole in it, and the hole opens and closes. When the hole is open, the proteins that form on it fold one way. When the whole hole is closed and you don't get a flow of warm water through it or whatever is flowing through it, the protein folds a different way. That's one of the ways that the same protein does different things. One time it might hold on to ATP, and in another folding type, or when it folds differently, it gets rid of the ATP or whatever. That hasn't been elucidated yet. It's just an idea. But we've seen some evidences of proteins folding in this whole ADP-ATP process. And for further information about it, it's in the Nick Lane book, if you happen to have a copy of it.
Yeah, I strongly recommend that you get a copy and read it. It's a really excellent book.
What's the name of the book?
The Vital Question.
Oh, okay.
And it is published by Norton Press, if that makes any difference to you. And if we weren't isolated, I would show it all to you, but maybe you can see that.
Yeah, we can. Oh,
You
it's
got it
down.
upside down.
Okay,
Yeah, yeah,
does that help?
Yep,
yeah.
great.
Okay.
I have another question. You brought up the reference to what we're going through now, the coronavirus, and you touched a little on that. And in this lockdown, one of the things I've done, among many, is to get to some of my old Scientific American magazines that have been hanging around and I've never had time to get to them. And I'm starting to read some of the articles. So I have the one, well, it doesn't have a date on it. It's just calling it anniversary of Apollo 11 is the, oh, here it is. I'm sorry. July 2019. And there's an article in there that I was reading just by coincidence, right in the same time period that we're going to have your lecture. It's called The Invulnerable Cell. And it's talking about recoding cells. Are you there?
I am there.
Oh, there you are. Okay,
Yeah.
I lost you for a moment. Recoding cells to be resistant to viruses.
Yes.
And I'm guessing that they didn't really go into it as to how they recode them, but it sounds to me like CRISPR. I mean, I don't know what else they would do.
It is CRISPR, that's what they're talking about. And
I wonder if, I mean, since we have a lot of time left and nobody seems to be asking other questions, if you could talk a little more about that.
well, this, I would sort of have to read up on it to know exactly what they're talking about. But CRISPR is a way of taking genes from another species even, and getting those genes incorporated into a cell of, say, us or of some other species. This can be done, and they're working on it every day, and it has become more or less a routine process. So what I would imagine they're thinking about is, for example, bats. Bats from which coronaviruses ultimately came are not particularly damaged by the coronavirus. We don't know why, but maybe there are certain genes that they have that allow them to harbor the virus in them. Maybe more, and I'm just thinking off the top of my head right now, is the idea that what kills you from a coronavirus infection is the hyperreaction of your immune system to that virus. It's not so much the virus infection itself, as I understand it, as the reaction that you're doing to the virus. Some of us, and nobody knows exactly which ones of us, hyperreact. We get the virus, we seem to be getting over it, and then all of a sudden we go downhill very fast. And the reason we go downhill very fast, apparently, is that we are overreacting to the cells that have the virus in them, and we are destroying those cells. So those people who overreact overreact in their lungs, because that's where the virus mostly is, and they end up damaging the lung cells of their own body because of this overreaction. So if I were doing this kind of experimentation, I would look at that process, that immune process that we all do. Is there some way of turning off the immune process in those overreactors? And there ought to be. And there ought to be a way of giving them the genes that people that don't overreact have. And I'm sure they're working on that, or at least I hope they are. Does that help?
It's
I don't know.
fascinating. It
I
helps a
don't know
lot.
if that's what the
I think
article
what this article is, as I recall from the article, is it's not so much attacking, addressing, developing a process to deal with the immune system as it is to develop a process where apparently when the virus enters the body, and we've seen the picture of the coronavirus with these little spikes all over it, and it attaches to I guess, a bacteria or a gene, whatever
it attaches to the cell wall of our cells.
Okay, and it needs a receptor to attach those little spikes to.
Yeah.
And so introducing what you've said is CRISPR, but the article doesn't say it, but it kind of can't be anything else, you know. Introducing then this foreign gene, they're trying to design it so that it will destroy or cut off the receptors inside the cell, so that when the virus arrives with its little spikes, there's nothing to attach to.
Yeah, yeah.
That's basically, you know, so the virus, I guess, dies because it can't attach and then replicate.
Well, that you summed it up very well, Lee, because all the viruses do that. Even these bacterial viruses that I was talking about, they have to attach. And once they attach, they push in their genes, through like a syringe. They just inject their genes into the cell, and then their genes take over the cell. But here's a problem that I would have to read the article. But the receptor that this coronavirus attaches to is the acetylcholine esterase receptor. And acetylcholine esterase is an enzyme that lung cells have, well, lots of other cells in the body too. People with high blood pressure have more of those receptors than people without high blood pressure. Apparently, the function of acetylcholinesterase is to moderate hypertension. When you are in your fight or flight mode, you are producing lots of things. They raise your blood pressure, that gets you organized to run away or whatever it is. And then that blood pressure has to come back down to normal. And that's what acetylcholinesterase does. But how do you block it? How do you block something that important to just the daily activities of the body? Maybe you can do that in the clinical setting. Anyway, I wouldn't approach it that way, but I'm not doing that research. Well, are any more questions? If not, I'm just so happy that you guys are still doing this, that in my limited knowledge of physics, I can even participate a little bit. And Bob, I just really have to thank you for that article you sent me, because it explained so much. And all we were talking about is elastic, rubber bands and fold.
Well, you're welcome. I just keep trying to find out more and more about what's going on. I'm going to stop the recording right now.